Doubting G, verse 11, speaks of the importance of what is not. So a pot is made of clay, but it's the inside that's useful. And a house is built with walls, but you live in the hollow space inside. And there's a similar situation going on in the desert, in that what you typically see in the desert are the vascular plants, but in between them, in the areas of what is not, there's a lot of things going on, including up to 70% of desert surface area being covered by these biological soil crusts, which are microbial communities in which the cyanobacteria bind soil particles together to create this living crust. And crust can be made of cyanobacteria, which is what I study, mosses and lichens. And the cyanobacteria are performing a ton of important processes, such as a lot of primary productivity, regulating nitrogen and water cycling, changing soil erodibility and pH. Here's a picture from my study site. You can see what this projector. The greenish stuff is the soil crust here. And given all these functions that soil crust perform, it's no surprise that they interact with vascular plants to a significant extent. These interactions can be positive or negative. They affect both the plant and the crust. But what hasn't been studied a lot is the pathways to interactions, which gets me thinking maybe the root system is an important pathway. After all, that's how the plant is interacting with the soil. My study system here is the Chihuahuan Desert grassland, which is what we have at the south. And two grass species, blue grama and black grama, co-dominate. And we know that they interact with soil crusts. But previous studies have focused mostly on early stages of the plant or soil responses, and haven't really looked at cyanobacteria fitness responses. And while we know that these interactions with plants can be species specific, there hasn't been a lot of research looking at differences between these two particular species, which is too bad because they're pretty ideal for such an experiment, in that they're quite similar in their above ground form and function. But they do have a different root system. Blue grama is a bunch grass and it has a more extensive root system, which means to me, perhaps, it's got a greater surface area over which the interaction can occur. So if interactions between the crust and plant is overall positive for crust fitness, we might expect to see more cyanobacteria near blue grama plants. Which informs my research questions here, looking at whether crusts are associating differently with one or other of the species. And secondly, I want to account for possible confounding variables of abiotic traits of the soil. Maybe crust is more dense when the soil is less coarse. To drive this point home, I have a high contrast slide to let you know that we're going to hunt down the answers to these questions one by one. And if we're looking at differences between plant species, we need pairs of guys. So, my mentor Annie and I set up 20 plots, each of which contain one black grama and one blue grama, focal individual. And then, of course, we needed some crust, which we collected by poking a little Eppendorf tube into the soil and getting maybe about half a gram in the tube. And we did that 5 times per plant. What we wanted to measure was cyanobacteria, but possibly a better measure is chlorophyll, because it can tell you how productive those cyanobacteria are, how much photosynthesis they're doing, how happy, fit they are. It's also a bit easier to measure. So what we did was we had to extract the chlorophyll from these cyanobacteria samples, which we did using DMSO. And then we looked in used a spectrophotometer to check the absorbance at wavelengths we know chlorophyll absorbs at, divided by the mass of the sample to give it density, and we found that there wasn't much of a difference between blue and black grama. And let me walk you through this mosaic plot. The numbers correspond to the plot number. The width of the bar tells you the total chlorophyll found in that plot, and then the purple is the proportion that was found near the black grama plant, and the gold is the proportion near the blue. And so you can see there's not really a trend per se. So what does that mean? Well, maybe the overall fitness impact of the interaction on cyanobacteria is neutral, or maybe the roots aren't an important interaction pathway. It's also possible that the blue grama's root system isn't all that much more expansive than black. A study found that a large percentage of blue's roots are actually within 5 centimeters of the base of the plant. So that could be another explanation. More chlorophyll data. First of all, you'll notice that on the y-axis it's around 0.5, which means that we're finding densities of 0.5 micrograms per gram, which means that the soil was 0.5 parts per million chlorophyll, which is pretty small. I was expecting to see numbers 7 or 8 times larger. Another strange thing is that we sampled on a day when it was very dry before it had rained this summer, and then on a day following rain. And chlorophyll density actually decreased after the rain. And I didn't know what to make of that. I thought maybe I did something wrong. I lost faith. But Lord Vader inspired me to do a little research, and I came up with a few possible explanations for this. One is that it was so dry that the raindrops were actually eroding the crust and decreasing productivity. Another possibility is that it was so dry that when it rained, the cyanobacteria cells started bursting due to osmotic pressure. And it's also been shown that high temperatures can combine with sudden moisture to slow the growth of or kill cyanobacteria. So what this could mean is, well, with climate change, it's getting hotter and rain events are getting less predictable, so that could be bad news for our cyanobacteria and all the other organisms that rely on the crusts. It also introduces the idea of a lag time in crust response to optimal conditions. It's arbitrary to assume that they're most productive when the soil is really dry, but I wonder how long being exposed to moisture before they are at their optimal production. With that, we will move on to the second question, looking at abiotic soil traits. What we wanted to do was create a model to be able to predict the chlorophyll concentration. And we were using these 3 soil variables and also plant species because why not include everything? And for moisture and temperature, the protocol is pretty straightforward. You just jam the soil probe into the dirt as hard as you can and then press a button, and it gives you a reading. What I want to point out is that before rain, moisture was 0, which really corroborates the idea of this erosion or osmotic cell bursting event. And for soil texture, which is actually 3 variables in itself, sand, silt, and clay, what we did was we drove the soil borer about 20 centimeters into the ground to collect 50 grams of soil, which I then measured out and suspended in a slurry and stirred it up, put in a hydrometer to measure the buoyancy after 40 seconds, and then again after 2 hours to get the rate of sedimentation, and plug those as well as temperature into some equations, and we get the soil texture in terms of sand, silt, and clay. But you might notice a problem with percent sand, percent silt, and percent clay, because once you know 2, you know the third one, right? It turns out all 5 of these variables are correlated, so you can't just wantonly plug them into a regression model and say that it's perfect. So what we did was we used principal component analysis to tease out and remove covariation between these variables. If you think of saying this as silt concentration, it only varies in the x direction. And then this is moisture. It varies in the y direction, but as you move along this vector of the variable, you're also changing with respect to silt. So with PCA, what you do is it creates 5 new variables out of the 5 that we have, taking bits and pieces of each one, and ranges them such that PC1 varies in the x direction, PC2 varies in the y direction, PC3 in the z direction, and so on into multidimensional space. I can't really draw that. The point is that when they're orthogonal to each other, covariance from one to the next is equal to 0. So what does that look like? The factor loadings tell you the magnitude and direction of how each variable went into that principal component. So you can see that PC1 was made up mostly of moisture, PC2 was mostly negative clay and sand. And also, we should point out the proportion of variance, which basically tells how important that particular principal component is. And what this means for us is that species 1 and 2 are explaining the vast majority of overall variation. So that simplifies the model a lot if you can not worry about species 3 through 5. So now instead of having 5 interdependent, weird soil variables and plant species, we have 2 totally independent variables, species, interaction effects. Here's the model. But the model wasn't very good. With an R-squared value of basically 0, that means that pretty much no predictive value at all. And in fact, stepwise model selection found the best model to be a flat line at the mean. Which, well, in a word. But I did find some cool stuff that I wasn't even looking for originally. When we ran multivariate analysis of variance of PCs 1 and 2 against plant species, we found that there were actually significant differences in the soils that these 2 plants were occupying. If you look at this graph, the red dots represent blue-gram individuals in their PC1 scores on the x-axis and PC2 scores on the y-axis. And you can see that there's kind of this cluster of them over on the left. And indeed, when you draw clouds around the center of gravity of those points, they're not really overlapping very much. And you might remember that PC1 was made up mostly of moisture. And yes, it turned out that black grandma was living in higher moisture soil, also higher temperature, and higher silt. And well, that rocked my world like a volcanic explosion because these guys are right next to each other. How is the soil that different? It's very cool. So what I take away from all this, first of all, the plot was categorically sandy clay loam, which was new for my study area. Previous researchers had always found it to be sandy loam or loamy sand. So that means that you've got high soil variability when it gets to flats. Also, you can see that blue and black grandma living in different conditions. Maybe black grandma is living in hotter soil that's also more moist, reflecting its tendency to start growing later in the growing season when temperatures are higher and there's been more precipitation. Blue grandma being in drier soil might be reflecting its 400-year lifespan versus black grandma's 40-year lifespan, in that it can better resist desiccation and it doesn't need as frequent periods of optimal conditions during which to reproduce. So what they're doing then is essentially blue grandma can specialize in the drier patches and lock down this mini niche and decrease competition between the 2 species. And then there's also potentially some climate change implications. The soil conditions are going to increasingly favor black grandma on this set, which climate change models predict black grandma will take over here actually, and the ecotone between the species is going to move northward. And that could be potentially unfortunate because black grandma is more susceptible to overgrazing, and it's not as good for erosion control as blue grandma is. With that, I just need to thank Annie, my mentor, for being incredibly helpful and knowledgeable and inspiring, and also doing a lot of labor as well. And Dr. Records and Beth and Eva for lab space and help in the field, and Dr. Sinsabov for letting us use the spec, and the NSF for funding, Amherst and the LTR for keeping everything running, UNM for the facilities, Kathy and John and everyone at Fish for letting me work on the SEV. And with that, I ask you, have you any questions?
Were the samples of the 2 different types of grass that you were still finding differences?